Hey guys, so like I said in my Edmodo post in my Remind 101, I'm holding you responsible for the cell organelles. That's something that you were taught your freshman year. It should be something that you can easily recall to memory. If you are struggling, feel free to use your book and figure out what the organelles are and figure out what they do. There are only about 13 to 15 of them. You should be able to do that without too many problems. This cell section is going to be fast and furious. Like I literally have it planned down to no more than 4 class periods, and that's including a lab and maybe even a test. We'll see. So tonight's video, or today's video, I don't know when you're watching this, is going to be over the cell membrane and transportation. So it's 2 sections in one video. So here we go. So as you know, your cell membrane is made up of phospholipids, and your phospholipids are obviously a lipid type of macromolecule. They're made up mostly of a glycerol head, also with a phosphate group in the middle, and then 2 long fatty acid chains. So if you look at the picture, here would be the actual molecule, and this is how we always draw it. This is the representation that we draw. Now, because of the phosphate group, that head is hydrophilic. It is attracted to water, it's water-loving. Whereas the tails, because they're all hydrocarbons, are hydrophobic. They're repelled by water. They don't want to be anywhere close to water. So because of these properties, these amphiphile properties, the phospholipids tend to arrange themselves as a bilayer, where their heads are in contact with water, here, and their tails are hidden, so all the water is forced away from the actual water. So if you look at it, see, this is why this section is white and not blue. So again, your water is going to be, here, and then here as well. There is no water here. All right, so again, it's one of those structure and function things. So this is what the whole arrangement would look like. Again, the polar region, or the hydrophilic region, is always going to be facing the water, which would be these heads. And the nonpolar region, the tails, will always be in the middle. Because of this, the whole membrane is impermeable to polar molecules. Because remember, the middle region of the membrane, the bulk of the membrane, is nonpolar. They repel each other. They don't like each other. So if you're small enough and you are not charged and you're nonpolar, you could fit through some of the spaces created when these molecules move back and forth. Because of this ability, the phospholipid bilayer serves as a barrier or a border. This is where the cell begins and ends. Nothing beyond this point without us allowing you through. So the cell membrane defines the cell. It separates the living insides of the cell from the aqueous environment that the cell is found in, so the liquid in your body. The membrane itself is really, really small. It's only about 8 nanometers thick, and it's kind of like, I always think of it like a bouncer in a club. It controls what comes in, and it controls what goes out. Some substances get to easily come in, and others not so easily or not at all. So if you're a cute girl, you get to go in the club, and you don't have to pay sometimes. If you're not that cute, or your outfit sucks, or you're causing trouble, you get to stay outside. So let's talk about its permeability to polar molecules. We know that because of those hydrophobic tails, anything that is naturally polar is not allowed through. But polar substances do come in and out of the cell. They do so because of the presence of membrane proteins, proteins that have inserted themselves periodically in between phospholipids. So if you look at this picture, these would be your phospholipids here, and then these colorful sections represent proteins. These proteins are very specific to certain materials, and they will allow those materials to gain access either in or out of the cell if they pass through them. This is what it would look like in actuality. Hopefully you recognize those alpha helices that are characteristic of the secondary structure of a protein. So your cell membrane isn't just lipids, and it's not just proteins. It's a combination of both. So we have phospholipids, And in between those phospholipids, we have what we call transmembrane proteins that are embedded in between. It's these proteins that allow the membrane to be semi-permeable. Certain things can pass through if they fit with the chemistry of the protein. So proteins are pretty much the perfect macromolecule to build these particular kinds of gateways or structures. And it's because those amino acids that make up proteins come in polar, nonpolar, acidic, basic, electrically charged, and all of those things. So every region of this protein can interact with the different parts of a phospholipid, be it a hydrophilic region or a hydrophobic region. So for transmembrane proteins, and transmembrane means that the protein spans the entire membrane. It goes from the outside all the way to the inside. It's going to be made up of a polar region that's going to interact with those heads, and a nonpolar region which will interact with the tail sections of your phospholipid. When it is within the membrane, it's going to be nonpolar amino acids. They're going to be hydrophobic, and they're going to help to anchor or hold the protein into the membrane. When we're talking about the outer surfaces of the membrane, so the parts that's actually in fluid, they're going to be polar amino acids, they're going to be hydrophilic, and they're going to extend into either the extracellular fluid, that's the fluid outside of the cell, or into the cytosol, the fluid inside of the cell. So to use the picture to help you, this section of the protein is considered within the membrane. So it is all going to be hydrophobic to allow it to stay in contact with these hydrophobic tails. This region, this purplish region, is the section that is polar. And that's the part that is going to be in the water outside of the cell or in the water inside of the cell. So here are some examples. One of the classic examples of a transmembrane protein is something called an aquaporin. And exactly as the name suggests, it is a water pore. It is a channel protein in which water can easily go in or go out of that particular cell. You see tons of them in bacterial cells. You see tons of them in any kind of cell that lives in a very aqueous environment. Some proteins can just easily let things slide on in, and it all happens through diffusion and there's no energy required, while other proteins aren't that simple. But we'll talk about that more as we get deeper into the video, especially when we hit the transport section. So what are the functions of these membrane proteins? We know they exist, but why do we have them? They have 6 major functions. The first one is to act as a transporter, which is what we've been talking about. It's a gateway or a channel or a tunnel to allow things into the cell that can't naturally diffuse through the phospholipids. Another reason is for enzyme activity. Sometimes these proteins themselves are enzymes and they produce a product of some sort. Another reason is to be a cell surface receptor. So when a beta Langerhans cell sends a hormone in the form of insulin to... Sorry about that. So when a Langerhans cell, which is the cell that makes insulin, sends its little hormone signal in the form of insulin out to the cells in your digestive system, something on that cell needs to be there to anchor or to dock or to receive the message that insulin is bringing, and then start a chain reaction, spread that message into the cell, telling the cell how to behave. So that's what a cell surface receptor is. Some of them are cell surface identifiers or cell surface markers like antigens. Have you ever wondered why your white blood cells don't digest everything inside of you? It's because all of your cells that are made by your body have special little markers called antigens, and it helps different cells to figure out, oh, this is a nerve cell, oh, this is a stomach cell, oh, this is a whatever cell. So it helps them to identify each other. Another use is for cell-to-cell adhesion. There are times when 2 cells need to be in contact with each other, but seeing as how they don't have arms and they can't literally wrap themselves around each other, they have one type of protein that is going to anchor, this would be our anchor down here, another type of protein on a different cell or on the same cell. I didn't do a good job of that. Hold on. For cell adhesion purposes, sometimes cells need to come together to communicate, to exchange information, or for reproductive purposes. So to hold the two cells in place, you're going to need an anchor cell, which is what an anchor protein, which is what this protein right here is going to do. You're also going to need a protein that is going to fit into the anchor, which is what this protein right here is for. So when the two of them come together, they kind of adhere and they allow the cells to stay in contact for however long they need to be touching. The last reason is it serves as a site, these proteins serve as a site of attachment for the cell's cytoskeleton. So each cell, even animal cells that don't have necessarily a specific shape, do have a cytoskeleton. Your cytoskeleton is important because a lot of your major organelles move around because of the cytoskeleton. They're anchored to the cytoskeleton. Think of it as the wires that the DART train runs on, that the DART train gets its electricity from. That's kind of how the cytoskeleton is for some of these organelles. But the cytoskeleton itself needs to be anchored because if it isn't, it'll just float off into the cell because the cell is mostly liquid. So some of these proteins act as attachment sites for that cytoskeleton, sorry. Again, all of these are in your book. So if you write all the names out, when you're done with the video, you can go to your book and get down, the definition or the explanation. Or you can just pause and write what I'm saying. It's completely up to you. Okay, so because there's so many different kinds of proteins, and because there's so many different types of cells, your proteins are going to determine how your cell's membrane functions. All right, and it's because we have tons of proteins and we have tons of cells. Each cell isn't going to have the same kind of proteins on it. We have classes of membrane proteins. The two main classes are peripheral proteins, which means that they're only on one side of the membrane, and then they're integral proteins, which means that they go all the way through the lipid bilayer. So again, peripheral proteins are loosely bound, and they're only bound to one surface of the membrane. So they're found at the top section of the membrane or the bottom section of the membrane, but they won't be all the way through the membrane. Integral proteins are found all the way through the membrane. They penetrate the entire layer. And examples of these would be those transport proteins that we talked about, those channel proteins, and they go from the top all the way through to the inside. All right, so here's a picture of the complete cell membrane with all its parts and pieces. And you'll realize that it's not just the thin line that we always draw to represent it. It's very dynamic, it has lots of parts and pieces, and they all serve a function. So these pictures are in your book, but it's good for you to see this, and you need to be able to identify what these things are and what their jobs and roles are. Now, attached to some of your proteins, you're going to have carbohydrate chains. You should have recognized them by their monomers of glucose, here and here. Some of these carbohydrates are also attached to actual phospholipids. When that happens, the name changes. So, let me change my ink real quick. So when you have a carbohydrate attached to a protein, this protein becomes a glycoprotein. Okay, so glyco from glucose. And when it's attached to a phospholipid, want to take a guess at what it's called? Yes, it's called a glycolipid. Okay, so what's their job or function? It's all about cell-to-cell recognition. Because of these macromolecules being attached to these lipids and to these proteins, they allow one cell to distinguish another cell. Okay, we can tell the difference between the two. These are what forms our antigens. These become really important when we start talking about organ and tissue development, and they're also the basis on which your immune system works. If your immune system doesn't recognize this antigen, it is going to digest this cell because it is going to think that it is something foreign and harmful. If it does recognize the antigen, then it will leave it alone and keep doing its thing. All right, so that's it for cell membrane. We're going to move on to cell transport. Sorry, obviously I was lying. We're going to move on to movement across the cell membrane. The same thing, still transport. So you should be really familiar with the concept of diffusion. And diffusion is based on the second law of thermodynamics. All biological systems are based on the second law of thermodynamics. And that says that the universe tends towards disorder or entropy. Things like to be in disarray. So the way we see that in diffusion is everything likes to be equal. We like to be all spread out. We like to be in equilibrium. And that's what diffusion allows molecules to achieve. It is the movement of molecules from a region of high concentration to a region of low concentration. So you're just moving from high to low, from a lot to a little bit. Simple diffusion, again, we're just moving from high to low concentration. It is a passive process, which means that we require no energy to do this. Now, this right here on my actual PowerPoint is a video or animation, and what you would see happening is the molecules will spread out. So they'll move until they're evenly dispersed. That's what you should see. I could probably play this for you in class when we get here. So diffusion refers to molecules. There is a special kind of diffusion that only refers to one molecule, namely water, and that is what osmosis is. So osmosis is the movement of water molecules from high to low concentration. So we've learned about passive diffusion. That's where no energy is required. The other type of diffusion is something called facilitated diffusion. Facilitated diffusion is diffusion with help. So things are still moving from high to low concentration, but they're going through a very specific pathway or a specific route. So think of those channel proteins. Facilitated diffusion would be diffusion through a channel protein. We're going through a very specific area. We must pass this guy to get to where we're going. It's helping us to get in. So those polar molecules that can't normally diffuse through the phospholipids, they would all diffuse through a channel protein. That would be facilitated diffusion. It's still moving from high to low. It still requires no energy. So it's still a passive process. Now with active transport, so far we've talked about passive transport. What you need to notice is passive always goes with the grain. We're always going from high to low. We're not trying to do anything special. With active transport, we're going to need to move molecules in the opposite direction. We're trying to move them against the concentration gradient. So instead of going from high to low, this time we're going from low to high. This will require energy because the proteins that do these jobs are nicknamed pumps. And it's because they literally do take one molecule up, push it all the way across, and release it on the other side. They have to undergo a conformational change, a shape change. And once we're doing all of that kind of activity, we will need ATP for it to run. So ATP runs the pump. Hold on. Okay, I'm back. So like I was saying, once we're undergoing a conformational change, and we're moving this time from low concentration to high concentration, there is going to be an energy cost. And we're going to have to pay that cost in the form of ATP, the energy molecule, the energy currency of the cell. So there are many different ways and many different mechanisms and models for active transport. You're probably not going to see molecules that look exactly the same, doing the exact same thing. Again, this is an animation that I'll play for you in class, but it gives an example of how there are different models for active transport. This particular model is called an antiport, and it is able to take in two different molecules at the same time. It can take in molecules of sodium, and it can take in amino acids all simultaneously and release them in two different directions. So the sodium goes in and the amino acids go out, or however, the amino acids go out in this case. This one's called a symport. And in a symport, you're going in just one direction. You still can take multiple molecules. Again, we're taking sodium and a different kind of amino acid, but they're all going to be pushed in one direction, either into the cell or out of the cell. So antiport, we can release molecules in two different directions, in and out of the cell. Symport, we can only release in one direction. Either they both go into the cell or they're both going out of the cell. But both of these would require ATP. So let's go over it again. So there are two kinds of passive transport, simple and facilitated. Simple diffusion is just a diffusion from high concentration to low concentration of nonpolar hydrophobic molecules, like lipids. Facilitated diffusion is still passive, but it's a diffusion of polar hydrophilic molecules, and they're going to do so through a protein channel, but they're still going from high to low concentration. Active transport is diffusion against the concentration gradient. So this time we're going from low to high. It is going to require the use of a protein pump, and that protein pump is going to require energy in the form of ATP in order to run. So here's just another visual, simple diffusion, small molecules that are all nonpolar. Facilitated diffusion, again, still small molecules, but this time they're polar. And active transport, various sized molecules, but they're going from low to high, and they need energy to do so. So, for the most part, we've been talking about really small molecules coming in and coming out of the cell. What happens when the cell has to move large molecules? It can do that as well, but it does it through a very different process. Cells are going to move large amounts of molecules or large individual molecules either through vesicles or through vacuoles. You should know what both of these are since they're both organelles found in the cell. There is one process that brings things in. It's called endocytosis. It breaks down into two sub-processes. And there's one process that takes things out called exocytosis. Endocytosis, like I said, is broken down into two smaller sub-processes. The first one is called phagocytosis, which is cellular eating. So think of a white blood cell engulfing a bacterial cell. That would be an example of phagocytosis. The other is penocytosis, cell drinking. And this is what the cell does when it wants to take in large amounts of very, very small molecules. This is more for one really large molecule. So few but big molecules, many but small molecules. And exocytosis is just how we get rid of it, how we push it out. All right, so let's look at it again. Here, phagocytosis. This would be penocytosis. There's another kind of endocytosis called receptor-mediated endocytosis, but it's only triggered by special receptors. So if those receptors aren't present, this process won't take place. Now, penocytosis is nonspecific. Once the molecules are small and the cell wants it, it'll take them in. Phagocytosis is kind of nonspecific, but something special is going to happen here. In order to digest this large amount of material, a lysosome, which is kind of like the stomach of the cell, is going to have to fuse with this vesicle so that digestion can take place. These molecules tend to be small enough that the cell can just absorb them. All right, so we know that water is special. Let's talk about how water moves across a cell membrane, because it's not very clear-cut. At least it's not always clear-cut. So like I said before, osmosis is just a diffusion of water. And we're still going to go from high concentration to low concentration, but it's going to happen across a semi-permeable membrane. So just like our cell membrane. Now, the direction of osmosis is determined by how much solute you have dissolved in the particular environment. The more solute you have, the less water you have. The less solute you have, the more water you have. Don't forget, it's the water that's moving, not the solute. And water is always going to go from high concentration to low concentration. So there are 3 different environments that your cells can be placed in based on the amount of solute. A hypertonic solution is where you have more solute and you have less water. So like 90% sugar and 10% water. A hypotonic solution is where you have less solute and more water. So 10% solute, 90% water. And an isotonic solution is where you have an equal amount of solute and an equal amount of water. 50% solute, 50% water. Again, water is going to move from high concentration where there's lots of water to low concentration where there's a little bit of water, but it's based on how much solute is present. So if you look at this example, here we have a lot more water, a lot less solute. Here we have a lot more solute, a lot less water. There is more water on this side. Water moves from high to low. So this would be high. This would be low. Water will move in this direction. Okay, so again, it's just based on solute. So why do we care? We care because our cells exist at 75% water nearly 100% of the time. And if we're not careful about our liquid environments and the amount of solute that they have dissolved in them, certain things could happen to our cells that would be detrimental. You have to keep in mind that animal cells are going to behave differently to plant cells. So when you put an animal cell into a hypotonic solution, don't forget hypotonic is where there's only a little bit of solute, there will be more water, more pure water on the outside of the cell than on the inside of the cell. So water will move into the cell. Eventually the cell will swell, and if water keeps coming in an animal cell, it would lyse. Lyse just means it bursts apart. This is bad. This is very, very bad because it means that your cell is now dead. When you put an animal cell in an isotonic solution, it is going to lose and gain water at an even rate. Okay? So it's going to stay a normal size. Nothing special is going to happen to it. You take the same animal cell and you put it in a hypertonic solution. That's one where we have a lot of solute in the water and a little bit of solute in the cell. There's going to be more water in the cell, so water will move out of the cell. The cell will start to shrink, and eventually it will shrivel. This also can be bad for your cells. Now, plant cells are a little bit different. Oh, and I'm sorry, an example of a hypotonic solution, that would be fresh water. An example of isotonic solution, like distilled water, because it doesn't really, it's not distilled water, but ionized water, because it's going to have the same amount of solutes dissolved in it. And an example of hypertonic solution, that would be salt water. Now we take our plant cell and we put it in the same 3 environments, and some different things are going to happen. Your plant cell actually isn't happy, or it isn't its most happy, unless it's in a hypotonic solution. Again, water is coming in, but there's going to be no lysing because plant cells have this really nice, strong cell wall that surrounds their cell membrane. The cell will only expand as far as the cell wall allows it to, so it will never burst. Plant cells like this environment best because their cells become nice and turgid and the whole plant stands up straight. This is normal for a plant cell. You put a plant cell in an isotonic solution, so like tap water, and you're gonna lose and gain water at a steady pace, but this is where your plant cell becomes flaccid, so the plant starts drooping over. It's not very happy. You put your same plant cell in a hypertonic solution, so salt water, and it's going to plasmolize because the same thing happens as with the animal cell. There's going to be more water in the plant cell. It's going to lose water continuously until most of the water is gone, and you're going to get this effect, this shriveled up effect. So how do cells manage their water balance if there are all these different situations? So this is pretty much what I just talked about. Again, hypotonic solution, your cell is in fresh water. There is a lot of water around the cell. This means the cell is going to gain water, it's going to swell, and it can even burst. How do we solve this? Now, there are some organisms that live in this situation all the time. We have a lot of microbes that are freshwater microbes, like Paramecium, for example. It means they're continuously taking in water. How do they deal? They have a special vacuole, a special organelle that deals with excess water. It is called a contractile vacuole. So as the excess water comes in, the vacuole soaks that water up, and then when it can't hold anymore, it pumps the water out of the cell using ATP for energy. So that's how the animal cell deals with the hypotonic solution. In plant cells, they actually want this scenario because it makes them turgid, which makes the cells full, which makes the plant stand up nice and straight, and we don't have a problem with bursting because the cell wall will prevent them from bursting. All right, so this is what Paramecium looks like. This is their contractile vacuole. This is what it looks like when it's full of water. And this is what it looks like when that water has been pumped out. So what about a hypertonic solution? This is a situation where your cell is going to have a low concentration of water around the cell, so water will come out of the cell because water is moving from high to low. This means that your cell is going to lose water and it could potentially die. So think of anything that lives in the ocean, like shellfish, for example. How are they going to prevent all of their cells from just pouring water out into the ocean? Their solution is they take up water or they pump out salt. Either they drink in more water, like they literally gulp it in, or they have special pumps in their cells and special adaptations where they will pull the salt out of the water and expel the salt, sometimes in their urine, sometimes in their poop. Some of them will just like push it out of their bodies, like around their nose or their ears. You'll see salt crystals forming. Plant cells, on the other hand, will plasmolyze, which leads them to wilt. Now, because of this wonderful cell wall, if you put water in, this cell will recover. So that's why even when plants look kind of droopy, if you water them, they'll spring back up in a couple of days. So that's how they deal. Sorry, that was Sigmund. Okay, so what about an isotonic situation? So think of an animal cell that's immersed in a mildly salt solution. There's not really going to be an issue because you're losing and gaining water at the same rate. There's no difference in concentration of water between the cell and its environment. So there's no real net movement of water. We're not losing water in either direction. It's exchanging equally. So in this case, for an animal cell, your cell is in equilibrium. There is no problem. The volume of the cell is stable. So think of your red blood cells in blood plasma. That's going to be an isotonic solution. Now, in a plant cell, it's not the happiest it could be, but can it live? Yes, it can. Right, we talked about aquaporins already. It's just specialized proteins that allow water to come in and out of cells. Yeah, these are the guys who figured them out. Nobody special. Moving. All right, so this should be it. As usual, come with your questions or comments. We'll take about 10 to 15 minutes and go over it in class. And you should be good to go. See you guys soon. Bye.